So there are different species of Acanthamoeba. More than 20 species have been described. Some of them are pathogenic, but not all of them are pathogenic. You can see some of the pathogenic species here, which include Acanthamoeba calvertsoni, castellani, and other species, and they cause different conditions. For instance, they cause a very fatal condition, inflammation of the brain, which is called granulomatous amoebic encephalitis. It's a very fatal condition and almost certainly fatal in people who get infected with Acanthamoeba. And this particular Acanthamoeba is also causing infection of lungs and skin in immunocompromised individuals. From immunocompromised individuals, we mean people who have problems with their immunity. For instance, people who are infected with HIV, where the T cells, they have problems with their T cells, or people who have some other autoimmune conditions, or people who have received some kind of grafts or transplants. They are always on immunosuppressive drugs. That's why they also are included in this particular category of immunocompromised patients. So these particular Acanthamoeba would cause these very fatal conditions in case of these individuals. So they should be very cautious because they are present almost everywhere. And in healthy individuals, it is causing chronic amoebic keratitis, which is also a very fatal condition. It's inflammation of the eye, and we would discuss this particular condition in this presentation as well. And the other conditions would be discussed in the following presentation. If you look at the evolutionary history of Acanthamoeba, um, you can see it here. This particularly, you know, this is a kind of scale which is defining different moments during the evolutionary period of eukaryotic cell. The Earth, which appeared about 4.5 billion years ago as a result of the Big Bang, and then there was origin of life between 4 and 4.5 billion years ago in the form of algae, bacteria, and bacteria which appeared on the surface of Earth, and you know, there were different events. We are not supposed to go into the details of those events. Here, our focus is just the appearance of amoebas or acanthamoebas. So, by looking at evolutionary history of amoebas or acanthamoebas, and by looking at the sequences and phylogenies that based on sequences of these amoebas, we can put them their appearance era in between the plants and fungi, certain types of yeast, which would be about, between 1 and 10 to the power 9 to 1.2 and 10 to the power 9 years ago. So it was probably during this era when the plants and fungi, they actually were diverging from the main eukaryotic cell lineages. During that particular period of time, in between these two eras, uh, at some point of time, these amoebas also are thought to have diverged from the eukaryotic cells, lineages, other lineages like plants and animals. Discovery. They were first discovered in the 1930s as a contaminant of the yeast culture. Cryptococcus pararhosis was cultured by some scientists, and they noticed that there was something else in the culture, and that was actually Acanthamoeba. It was later placed in the genus Acanthamoeba and then described as a causative agent of Acanthamoeba granulomatous encephalitis, or AGE, in the 1960s. And after some times in the 70s, it was isolated in case of patients who were suffering from keratitis. That's why, it was then, declared to be a pathogen, causative agent of the keratitis condition or inflammation of the eye. Um, by looking at the biology of this particular parasite, uh, acanth is a Greek word which means spikes. You can say that it's an amoeba with spikes. Spikes are very popular these days because of the coronavirus spikes that people are mm, talking about. So we're very scary and fearful structures, and the spikes are not new to microorganisms. They are present on microorganisms that even had emerged in the past, just like this one. But these spikes definitely are different from the viral spikes, but they got their names because of the spikes, Acanthamoeba. And these are typical eukaryotic cells. They have specialized structures, membrane-bound structures, just like different vacuoles. Different vacuoles are like large contractile vacuoles for osmotic regulations. They have digestive vacuoles. They have glycogen storage vacuoles, and they have lysosomes. All these structures are membrane-bound. And they have a double layered membrane which is bound by two layers, just like in case of other eukaryotic cells. And normally there is one, but can be many. In some cases, people have seen there are more than one nucleus inside there. And mm, if you look at the volume that the nucleus would occupy inside a trophozoite or inside the cell, this parasitic cell, it's about 1/6 of the entire volume. And if you look at the composition of the plasma membrane, it also is, more or less the same as you would see in case of other mammalian cells, including animals or human beings. With proteins, 33% of it is protein, phospholipids, sterols, and lipophosphonoGlycans, which are 29%. So if you look at the composition of the plasma membrane, you can guess that the plasma membrane would be a very functional structure. There would be integral proteins, and there would be proteins responsible for different functions, just like acting as channel proteins, as receptor proteins, and performing different types of functions as you would see in case of other eukaryotic cells. And similar is the case with different phospholipids. And there are also different mitochondria, lots of mitochondria inside. So there are also chloroplasts inside. So these specialized structures are performing different specialized functions, and you know uh, these specialized functions actually are making it very unique in a sense that sometimes you would take it for an individual rather than an individual cell. You would take it for an individual organism. It's a full-fledged organism, actually, it's specialized structure. And the genomic size is variable. Different species, they vary in size from 41 to 45 megabytes. And there are 17 different genotypes which have been discovered so far. Genotypes are named from T1 to T15, T1, T2, T3, up to T15. And some of the genotypes are very pathogenic, like the T4 genotypes, which is responsible for 90% of the keratitis or inflammation of the eye. The T1 genotype, it infects the brain tissue only. It means it causes the encephalitis conditions. There are some certain strains are the T1 genotypes which are causing this particular condition. And the T4 genotypes, it has the potential to infect all many different tissues like the skin. It can infect the skin and cause this ulceration. It can infect the brain and cause this inflammation. Also, it can cause inflammation of the lungs. So different genotypes, they have different preferences for tissues, and some of them they can affect many different tissues at the same time. Uh, the habitat, they are ubiquitous. We would discuss this particular aspect in the upcoming slides when we discuss their, diversity of this particular, the diversity of uh, the pathogens and the diversity of different environments where you can find this particular pathogen. uh, you know so that would be discussed in detail there, but you here shortly we can say that it is present almost everywhere on this planet Earth in many different extreme environments. You can find this particular parasite. And if you look at the morphology of this parasites, it is found in two different forms: the trophozoite form, which is the active form, and the cyst form. And these more than 20 species of this particular parasites, they are divided into three main groups based on their size and cyst morphology. The cyst morphology is important for this grouping. They are divided into three main groups based on this morphology of the cyst. So as far the trophozoite is concerned, trophozoite also is extremely variable in shape and size. There is no particular shape, or you can not say it does that it has got a single shape, or that you can just say it's round or cuboidal or some other shape. So it keeps on changing. It has variable shapes, and its diameter, it also ranges from 10 to 40 micrometer. The cytoplasm of the trophozoite is finely granular, and there is a single nucleus with a large, dense, central prominent nucleolus surrounded by a halo. Acanthamoeba, slender spine-like projections of plasma membrane are there that you can see in case of the trophozoite. So it's a trophozoite. You can see it's an electron micrograph of the trophozoite. It's an active and infective stage of this particular amoeba. Uh, it is active as well as infective, and it's the feeding stage. It feeds on different types of bacteria, fungi, and other microbial species. The optimum temperature for you know uh, the parasite to transform into this trophozoite form is 30 degrees Celsius. It requires a neutral pH, and it also requires nutrients. If all these conditions are there, then it would be converted into a trophozoite. If the conditions are not good, then of course it would be kept in another form or changed into the cyst form. It feeds on single-celled organisms just like the mycobacterium, avium, etc. And these bacteria which are residing inside this particular trophozoites are not only, although the relationship is not very clear that what benefit uh, uh, you know uh, is there for these species to live inside these host organisms. So they have been experimentally observed to multiply inside this particular host. So the host of course, the parasite is getting something out of it, but what the bacteria is getting out of this relationship is not clear. but it's clear that some of these bacterial species are multiplying inside this particular parasite. The cyst, varies in shape and measures about 15 to 20 micrometer in diameter. It's covered by a double layer cyst wall. Outer wall are ectocyst, it's wrinkled or rippled in shape, and the inner wall or endocyst, which is smooth and has spores or opercula. And this opercula or pores are for an excitation when it has to come out of the cyst and become active again when the conditions are favorable. Central nucleus with a large dense karyosome surrounded by clear halo is seen inside the cyst. So cyst is capable of surviving lack of food sources, desiccation, extreme pH and temperatures, as well as ultraviolet radiations, among others. So these extreme conditions could be tolerated by the cyst form, which is covered by double-layered, cell wall. This is the cyst. You can see this is the rippled outer wall that you can see. Then you can see the inner wall. This is then the nucleus. You can see a halo around the nucleus and the dense material inside, and different granules and vacuoles that are visible inside the cyst form of this. So excitation, uh, you know, the nucleus, you can see the different forms. So this is the active form on the left side. On the right side, you can form this cystic form of the parasite, and this keeps changing. This, you know, uh, from the active to the cyst form, or from the cyst to the infective form, or the tropocyte form. These, uh, this, you know, change in the morphological form actually is because of these conditions that we have discussed. Availability of nutrients, the pH and temperature, all these things are important in this. And this is the slide that I wanted to show you. Distribution of this particular parasite in the environment. It is found in diverse natural environments. You just look at these different environments. I have not enlisted all, but sea and pond water, soil, freshwater lakes, hot springs, saltwater lakes, Antarctica. It is also found in the air. It has been isolated from mineral waters, from factory discharges and air conditioning units and sewage water, composite and vegetables, and kitchen sprayers, and from surgical instruments, and you can you name it. So many different sources that it has been isolated from. Fresh water from healthy, diseased, and dead animals, from hospital settings, from cerebrospinal fluid, and for different parts of the human being. So it's almost ubiquitous in the environment. You can say it's almost present everywhere in the environment. And people, can get infected if they come in contact with these pathogenic species that we discussed earlier. Not all of the species are pathogenic, but many of them. Even the pathogenic species have been isolated from many different settings. So if you have, if you receive some kind of injury to your eyes, and then you are washing it with contaminated water, then of course you can get infected with this particular parasite. It also can cause some airborne infections as well. Uh, if you look at the pathogenesis, that how mm, it causes different conditions under you know, unfavorable condition, as we discussed earlier. The trophocytes here at the bottom, under unfavorable conditions, would just, undergo encystation or cyst formation. And the cyst would look like this. Okay. And under favorable conditions, optimum temperature of 30 degrees Celsius, favorable pH, and the nutrients, in all these conditions, again the cyst would turn into the trophozoites. And the trophozoites then can infect human beings through the aerosols or through skin penetration. So if it is through the aerosols, the aerosols can get into the lungs, or the aerosol can get, you know, into your nose, and the olfactory neuroepithelium can also be infected with this particular parasite, thus leading to inflammation of the brain. And if it goes into the blood, into the lungs, for instance, when you inhale it with respiration, then after infecting the lungs, it can enter into your bloodstream, and the blood circulation, through the blood circulation, it can infect, you know, your brain tissue or other organs. Similar is the case with the skin penetration. The skin penetration can also lead to, you know, the parasite entering into the blood, and from the blood tissue, along with the blood, it would just, you know, go in infect the brain tissue. How does it crosses the blood-brain barrier? It's not very clear. It's, you know, people are still struggling with it, doing lots of different research in order to find out that how this parasite actually is crossing the blood-brain barrier. So the aerosol can get through the lungs or the nose, you know, and infect the brain with the help of blood or without the help of blood. And the trophozoites, after penetrating into the skin, can also get into the blood and then be travel to the brain tissue, and where there it would cause inflammation of the brain, or AGE, or maybe you know, glomerulomatous encephalitis. So this is a condition, Acanthamoeba keratitis. You can see it here. It's a normal eye at the bottom. It is Acanthamoeba infected eye. This opaque structure that you see is because of infection with this particular parasite. So it can cause complete blindness in people if the condition is not treated. There are different treatments, and there are surgical procedures which are used for the treatment of these particular conditions. So in healthy individuals, we are not talking about immunocompromised individuals here. We would talk about immunocompromised individuals in the next lecture. In healthy individuals, it causes this particular condition and inflammation of the eye. And it is seen most particularly in individual who uses contact lenses, or who receive some kind of trauma or injury to the cornea of your eye. In these individuals, this particular condition has been observed to be more frequent. And it happens so because, uh, as we talked about the distribution of this parasite in the environment, it is ubiquitous. It is present almost everywhere in the environment, in all kinds of water, whether it is drinking water or processed, highly processed drinking water, just like the commercial water that we buy for drinking, or tap water, or you know, other the water types. It's present almost everywhere. So it means that somebody can get infected very easily if you receive some kind of trauma to the cornea of your eye, and you know, or if you are using contact lenses for longer periods of time. Actually, this was this particular parasite was discovered to cause this particular keratitis or this condition. Uh, Initially, it was discovered, or it was probably in the 1970s that uh, it was associated with the condition was associated with this practice of wearing contact lenses because it was more prevalent in people who are wearing contact lenses. Uh, Later on, it was the parasite was isolated uh, from different settings, which confirm these observations or association studies. And it's a chronic progressive ulcerative disease. And the cornea shows characteristics annular infiltration and congested conjunctiva. If not treated, progress to corneal perforation, blindness, and loss of eye. You can completely lose on your eye and eyesight if you do not treat this particular condition. It's a very tough or very severe condition. If you look at the immunology, the first line of defense against this particular parasite is the tear fluid produced by lacrimal system in your eye, together with the constant eyelid movement. The eyelid movement, actually, it's a protective moment, and it's the first line of defense for your eye. This blinking moment, it's not only sequestering these lacrimal secretions or the tear fluid in your eye in order to wash out different pathogens. Uh, you know, uh, it is a kind of protective mechanism for your eye. And this tear fluid, which is produced by the lacrimal system, uh, it makes a tear film. That tear film has got three distinct layers, and you can divide these three layers into aqueous layers, mucus part, and there is an oily layer. So there are three layers: aqueous layers, mucus layers, and oily layers in the tear film, which is formed here at the surface of your eye. Cornea is uh, avascular. It's not avascular, it's avascular. Avascular means that there are no vessels in the cornea. So you can say that if there are no vessels, it's a kind of immunocompromised, or you can say that it's not very well protected as compared to other structures of the eye or other structures of your body because of no vessels or non-vascularization there. Uh, because vascularization is needed for well protection or good protection, because that particular structure which is vascularized will get blood, and the blood contains different types of cells and antibodies and different substances which can deal with different types of pathogens effectively. But if a structure is not vascularized, it means that it's a kind of immunocompromised, or its protection is compromised. So in the tear film, there are three layers, and aqueous, mucus, and oily layer. In the aqueous layer, it contains antimicrobial lysozymes, lactoferrins, and secretory IgAs, or SIGAs. These are present in the aqueous part of the tear film. So as this eye blinking moment actually is helping with the spread of these antimicrobial lactoferrins and secretory IgAs, on the surface of your cornea. And it is a protective mechanism which is protecting your eye against the pathogens. On the other hand, conjunctiva, it's highly vascular, you know. there are lots of vessels in conjunctiva. If you look at the, this diagram that I have included here, this is cornea, the outermost layer. Okay, this is pupil that you see here. This is the lens, the bigger one. The pupil is right here. You can see this bulging out of the lens, and this is iris. You can see these two lids on one side and another side, and the cornea. This is the optic nerve, and this is the retina here. Okay, so this is highly vascular, and conjunctiva is highly vascular, and immunoglobulin A producing plasma cells are also there in the conjunctiva. So when it is vascular, you can imagine there would be T lymphocytes and natural killer cells and macrophages and all these things in the conjunctiva, and which in turn means that cellular and humoral responses, cell-mediated immunity and humoral or antibody-mediated immunity, or these responses for the clearance of the pathogen would be initiated in conjunctiva. So it's hard for the parasite to reside in conjunctiva rather than residing on the surface of cornea, which is less protected as compared to conjunctiva. So in order to proceed uh, in order to proceed with infection, Acanthamoeba must remain at the corneal surface and invade into the stromal tissue. And the tear film in contact lens wearers differs in terms of volume and makeup, which alters the levels of inflammatory mediators in tears, thus causing increased inflammation. And the third thing is occlusion of atmospheric oxygen and thinning of the basal tear film, which is responsible for the metabolic compromise of the cornea and also lead to less resistance to microbial infections. Okay, so is uh, you saw here that conjunctiva is highly vascular. For that purpose, it would not be easy for the pathogen to, uh, you know, just jump into that particular environment. So it has to, if it has to proceed with infection first, it has to stay on the cornea for some time and invade into the stromal tissue gradually. So the tear film, what happens in case of the contact lenses wearers, that the tear film in contact lens wearers, it becomes thinner, uh, you know, and that would actually change the level of the inflammatory mediators which are present in the tear films. We talked about these inflammatory mediators, antimicrobial lysozymes, lactoferrin, secretory IgAs, etc. And that would, when the tear film or your protective response is under stress due to some reason, it means you are more prone to develop this condition. So this condition would be good for the parasite to prevail there and invade into the stromal tissue and just enhance this particular condition or deteriorate the condition inside the eye. Okay, and this thinness also causes occlusion of atmospheric oxygen, so it's a kind of metabolic compromise of the cornea as well, which leads to less resistance to the microbial infection. So this is what happens here in the case of. So once in the stroma, once Acanthamoeba is in the stroma, Acanthamoeba secretes proteolytic enzymes. And these proteolytic enzymes would carry about stromal degradation. And this stromal degradation will lead to infiltration of the macrophages and neutrophil infiltration would also follow. Because the degradation would lead to attraction of the macrophages would secrete cytokines which would attract more neutrophils to the site of infection. So this would be a kind of cell-mediated immunity against this particular parasite which would be instigated. Similarly, this particular response then would modulate the B and T lymphocytes activity to clear Acanthamoeba. So the cell-mediated immunity would be followed by humoral immunity, or modulation of the B and T lymphocytes for the clearance of Acanthamoeba. And this is what happens if the humoral response and cell-mediated response are stronger enough. Then the parasite would be cleared off. Otherwise, the parasite would just persist there and it would just, cause blindness in people uh, who are infected with this condition. So this is, I think, enough for this part of the lecture. And in the next part, we would discuss about the growth cycle, and we would also discuss about um, different types of pathogens, bacteria, viruses, fungi, etc., which are living inside these Acanthamoeba species. And when we talk about emerging and re-emerging pathogens, or the reservoirs of different viruses or different bacteria or different fungi, so these Acanthamoeba species or the parasitic species are very important from that point of view, because when we say that you do not know how many different species the coronavirus, for instance, currently would have infected, and where would this particular coronavirus, which is causing the pandemic these days, would be living, and in how many different types of species it would be living. So these parasites are important from that point of view as well. And this is also a very good lesson for the students to uh, you know design their research projects in light of this information, in order to look for different types of pathogens which are living inside parasites. So that would be interesting as well. And we also would discuss about this parasite uh, causing its involvement in other aspects, like the microbial uh, communities which are affected by this parasite in the environment, because it is feeding on different bacterial species and other fungal species and other parasites as well. So the balance of the microbes also in the environment also depends upon these, and availability of nutrients in the environment uh, uh, is also dependent on these amoebas or acanthamoebas. So we would discuss all those interesting aspects along with its use as experimental model to study the movement or locomotion in organisms. That part will also be discussed in the next, uh, presentation. So you have to go through the next presentation in order to be fully informed. Thank you very much.